Okay, so get this. Scientists have basically achieved a level of control over protein design that we've only really dreamed about until now.
Right.
Using deep learning, they've managed to engineer proteins that can dynamically, morph between multiple distinct shapes on demand.
Mm-hmm.
It mirrors those really sophisticated signaling mechanisms you see everywhere in nature.
And the really mind-blowing part, it's not just predicting these shape changes anymore.
No.
No, it's actually designing the movement itself. We're essentially programming function right there at the molecular level.
Wow. It's a programming movement.
Yeah, and what's truly significant here is, well, we're moving beyond that old era of just static protein design.
Right, the fixed structures.
Exactly. For years, the main focus was just creating single, stable structures. But the real challenge, and honestly where biology just excels, is in dynamic proteins.
The ones that move.
The ones that undergo controlled shape changes, yeah.
Yeah.
I mean, think about how many essential processes rely on this, like the activation of kinases through those subtle helix rotations.
Okay, yeah.
Or how GPCRs kind of kink their helices to relay signals across membranes. These natural molecular switches, they're now inspiring this whole new wave of engineered ones.
Exactly. And the traditional physics-based models, I mean, they're powerful, don't get me wrong, but they kind of struggled, didn't they, to accurately capture those really delicate energy landscapes that govern these transformations?
It really did. The energy differences are tiny.
And while deep learning has just revolutionized our ability to predict static protein structures with, frankly, incredible accuracy,
Unbelievable accuracy, yeah.
applying that same AI smarts to these, fluid, dynamic systems, that represents a major leap forward. It feels like it's opening up a whole new field of possibilities.
Absolutely. And this research, it tackles that challenge head on. The core idea is really powerful. Leverage the speed and, the pattern recognition of deep learning.
Right.
Combine it with the detailed insights you get from molecular simulations.
Ah.
And you can not only design these intricate intradomain motions basically from scratch, but you also gain a much deeper understanding of the actual atomic interactions driving them.
Okay, so let's dive into this groundbreaking approach they took. Figure 1 in their paper illustrates the central concept, right? Designing proteins that can exist in several, precisely defined conformational states.
Mm-hmm.
And it looks like their starting point was a protein already known to bind calcium ions.
That's right. They used this existing calcium-binding protein as a kind of scaffold, a foundation. The method, which they detail in Figure 1, involved a pretty clever series of steps.
The what?
Okay, so first, they performed what's called an in silico mutational scan.
In silico, so
Wow.
using computer simulations.
Exactly. A scan using simulations to pinpoint the absolute minimum set of amino acid residues, the building blocks that are crucial for defining each of their target shapes, their target conformational states.
Okay, so how did this in silico scan actually work? It sounds like they were sort of virtually tweaking the protein's code.
That's a good way to put it, yeah. They systematically checked how making small single amino acid changes, specifically changes that would make the protein sequence more similar between the different target states, how those changes would affect the protein's predicted 3D structure.
Ah, I see.
The goal is really to identify those key switch residues, the ones that, when you alter them, preferentially stabilize one shape over another.
Got it. So they found these critical control points, these switches. What was the next step?
So once they had this minimal set of key residues identified, they then focused their sequence generation efforts mainly on varying things at those specific positions.
Okay.
The aim was to create a whole library of related proteins where each one had slightly different preferences for the target states, all because of these subtle variations in those key spots. It effectively allowed them to sort of dial in different equilibrium distributions of the conformations.
And the result, I mean, it sounds like they actually designed protein switches that could flip between these desired shapes on a biologically relevant timescale.
Yeah.
They mentioned the low microsecond range.
That's right, which is incredibly fast at the molecular level.
Yeah.
Indeed. And this naturally leads us into Figure 2, because Figure 2 explores how the balance, the equilibrium between these different states can be precisely controlled, precisely manipulated.
Okay.
And here's where it gets really interesting, because they uncovered several quite elegant control mechanisms.
All right, I'm listening. What did they figure out?
Well, the first mechanism is maybe surprisingly simple, but incredibly powerful. Just single amino acid changes.
Just one change.
Just one. They found that by making just one change at one of those critical switch positions they'd identified earlier, they could dramatically shift the equilibrium. They could effectively tune the whole system, ranging from a state where the protein almost exclusively adopts one shape, all the way to a state where both shapes are present in roughly equal amounts. That's just an astonishing level of control from such a minimal tweak.
That really is a tiny molecular adjustment, but potentially huge functional consequences down the line.
Exactly right. Then, the second control mechanism they leveraged involved calcium binding.
Back to the calcium.
Back to the calcium. Remember, their starting point was a calcium-binding protein, so they ingeniously designed a calcium-binding site within the dynamic region of their new proteins.
Okay.
But, and this is the crucial part, they designed it so this site would only adopt a structure capable of actually binding calcium in one of the two conformational states.
I see. So the presence or absence of calcium itself acts like a kind of molecular lever, pushing it towards one shape over the other, like a built-in sensor and actuator.
Precisely. Calcium, which is, a super common signaling molecule inside cells,
Ubiquitous, yeah.
can then directly regulate the population of these states.
Yeah.
High calcium, one state favored, low calcium, the other. And they didn't stop there. They also looked into allosteric mutations.
Allosteric, right? That means changes at a site that's far away from the main action, but still influencing it, correct?
Exactly that. They showed that by introducing mutations at locations pretty far removed from the calcium binding site itself, they could still fine-tune the conformational equilibrium.
Huh.
And even alter how strongly the calcium binds. It's a beautiful illustration of how interconnected a protein's structure really is, how influences at one point can just ripple through the whole molecule.
Okay, so these computational designs and predictions are really compelling. But how did they actually check? How did they confirm that these designer proteins behaved like they expected them to in, a real lab setting? This is where the rigorous validation comes in, I assume?
Absolutely critical step. You can't just rely on the models. They performed extensive experimental dynamics analyses.
Okay.
And the results provided strong support for their design principles. But beyond that, they undertook the really challenging task of determining the actual 3D structures of 4 of their designed proteins using NMR spectroscopy.
Nuclear magnetic resonance, yeah, not easy.
Not easy at all. And these experimentally determined structures showed an exceptional level of agreement, like really close agreement, with their computationally designed states.
Wow.
It confirms they achieved atomic-level precision in their designs. That is some powerful evidence right there.
That really is serious confirmation that the whole design approach actually works in practice. And the story gets even better, doesn't it? There's more validation.
It does. They also found a strong correlation, a strong agreement between their deep learning predictions, the results from separate independent physics-based simulations, and all the experimental data they gathered.
So everything lined up.
Everything lined up beautifully. And this convergence of evidence was key. It allowed them to really dig into the nitty-gritty details and actually identify and even, in some cases, reprogram the specific networks of atomic interactions that stabilize each distinct shape, each conformational state.
Okay, so let's try and bring this all together then. What's the core message from all these findings? What's the key advancement here?
Well, I think the fundamental achievement is the validation of a general and seemingly robust method for designing proteins that have distinct conformational state shapes that are entirely specified by the designer beforehand.
Right. We decide the shapes.
Exactly.
Yeah.
Just like nature uses things like ligand binding and these allosteric mechanisms to control protein equilibrium. This work shows we can now engineer those kinds of capabilities pretty much from scratch. They've essentially created new ways for proteins to undergo controlled movements within the molecule, inspired by those really critical dynamic events we see in natural signaling pathways. It significantly expands our protein engineering toolkit.
It really does feel like a landmark moment in protein engineering. So if we step back and look at the broader picture, what makes this work so innovative? What's the leap?
Well, methodologically, I think this work fundamentally redefines what it means to design a protein. We're no longer strictly limited to thinking about just a single static 3D arrangement of atoms. These researchers have effectively added a time dimension to the design process.
The fourth dimension.
Kind of, yeah. It's a capability the field has been striving towards for a long time. It's about designing not just a structure, but a controlled dance of structures over time.
So it's not just capturing a single snapshot anymore. It's like choreographing the entire molecular movie.
That's a perfect analogy.
Yeah.
Precisely. And this obviously opens up just an enormous range of potential applications down the road.
Like what?
Well, imagine being able to design programmable signaling systems,
Yeah.
basically from the ground up. That could let us build much more sophisticated, more responsive biological circuits. The researchers themselves talk about the exciting possibility of engineering even more complex behaviors,
Mm-hmm.
like proteins that can integrate multiple different signals or perform coordinated movements that are actually driven by some energy input.
That sounds incredibly versatile. Are there other possibilities this unlocks too?
Oh, definitely. They also highlight the potential for taking existing natural proteins and engineering non-native new dynamic motions into them. That gives us a whole new way to precisely control their biological activities.
Okay.
And beyond the fundamental research, you can see this leading to things like tailor-made biosensors
Right.
that
No.
respond to specific environmental cues just by changing shape, or controllable therapeutic proteins that only become active under specific conditions, say inside a tumor, but not elsewhere in the body.
Targeted activation.
Exactly. And even sophisticated nanoscale machines with actual moving parts.
Mm.
Think about like force-activated biochemical valves or maybe enzymes that change their activity level in response to mechanical stress.
It really feels like we're shifting gears, moving from designing, I don't know, inert building blocks to designing actual functional machines at the molecular scale.
That's a great way to put it. Functional machines.
So looking ahead, what key insights from this specific work could really pave the way for future research in this, frankly, super exciting area?
Yeah, it is exciting. Several key insights stand out, I think. First, that synergistic combination deep learning plus the detailed insights from molecular simulations that looks like a really powerful strategy, both for designing and for understanding dynamic systems.
Okay, the combo approach.
Right. Second, their innovative use of those in silico mutational scans to pinpoint the crucial residues dictating state preference. That provides a valuable blueprint, a methodology others can adopt.
Makes sense.
And third, just the remarkable ability they demonstrated to precisely tune the conformational equilibrium, that balance between shapes, through just minimal sequence changes and those distant allosteric mutations. That offers a level of fine control we just haven't really had before.
Right.
Ultimately, I think this work convincingly demonstrates that AI, artificial intelligence, can go far beyond just predicting a single protein fold. It can actually specify an entire energy landscape, the collection of different shapes a protein can adopt, and the probability of it being in each one. That's a fundamental shift in our capabilities.
So just to summarize the impact then, this research really feels like a major, major step forward in our ability to truly engineer protein function. We're not just predicting static structures anymore. We're actively designing proteins with built-in movement and, crucially, the ability to dynamically respond to their environment.
Exactly. It's a clear transition from designing static components to creating active, responsive molecular machines. It's a whole new level of engineering.
Okay, so here's maybe a final thought for you, the listener, to chew on. Now that we actually have this ability, this power to design proteins that can switch shapes on demand with such incredible precision, what entirely new kinds of biological circuits or nanoscale machines could we potentially build? I mean, could we be on the verge of creating completely novel forms of biological regulation? Or maybe even finding unprecedented ways to interface with the cellular processes already happening inside us? It feels like a profound leap that, well, it opens up a universe of possibilities.